Bismillahirrahmanirrahim. Today we are going to study about how the coronavirus vaccine works. There are many types of coronavirus vaccine, and in this tutorial, in this first lecture, we are going to study about how the Pfizer's and the Biontech corona vaccines work, and how the Moderna's coronavirus vaccine works. So in this tutorial, we are going to study about these two types, and in the next lecture, we are going to study about the other types of coronavirus vaccines. So let's start. So first of all, talking about the basics of vaccine. So how do the vaccines work? How they work? So they works when we give a small amount of harmless substance in the palm of live attenuated, i.e. weak virus or other organism like bacteria, or we may give it in a palm of killed virus or bacteria, and in the palm of antigenic protein of that virus or bacteria. When we give these kind of substances or inject these kind of substances into the body, so then the body sends these substances as foreigner and produce antibodies against it. And then these antibodies encounter and prevent us from the disease by neutralization of the disease causing organism, or by complement formation or activation. They kill the substance, or by any other way like opsonization, which helps in phagocytosis of the organism. Now talking about one of the two most important vaccines against the coronavirus COVID-19, which is the Pfizer and the Biontech vaccine. So its official name is BNT162b2, and its generic name is Tocilizumab, and sold under the brand name of Comirnaty, having an efficiency of about 95%. Now talking about the second vaccine, which is Moderna vaccine, and it is officially called as mRNA-1273. Now to understand the working of coronavirus vaccine, we have to study about the important properties regarding the coronavirus. So first of all, talking about its genome, so it has a non-segmented single-stranded positive polarity RNA, and this non-segmented single-stranded positive polarity RNA encodes for some of the proteins, in which one is the RNA polymerase, and this RNA polymerase is required for the replication of its genome, And the other protein which is important regarding the coronavirus vaccine, it is a structured protein called spikes. And these spikes are present on the surface of the virus, and through these spikes, the virus attaches to the cells and cause disease. Through these spikes, the virus attaches to the cells and cause disease. So now talking about the basis on which this Moderna virus and the Pfizer and Biontech virus is based. So this virus is based on the virus genetic material, i.e. messenger RNA, and this messenger RNA encode for the spikes. So this virus is based on the messenger RNA, and this messenger RNA codes for the protein which is called spikes, and these spikes are present on the surface of coronavirus like this. And due to these spikes, the coronavirus is called coronavirus, because the corona is derived from the word cron, i.e. cron-like, having spikes. So this virus is coronavirus. So this coronavirus vaccine is basically a messenger RNA. So now here comes the problem that injecting a messenger RNA predisposed to the natural viruses in the body. So in order to carry them safely inside the body, because they are very fragile and could break down and choked by the natural viruses, so they are wrapped in the oily bubbles. So they are wrapped in oily bubbles to protect it from the natural viruses in the body. For example, this is an oily bubble you can see, and this is the messenger RNA in which it is wrapped. So now talking about the mechanism that how this coronavirus vaccines work. So when we inject this this coronavirus vaccine into our body, this coronavirus vaccine attacks our cell and cause fusion between this coronavirus vaccine and the cells. After fusion, the messenger RNA enters our cell. Messenger RNA enters our cell and is translated into the spike protein by the ribosomes inside the cell. This spike protein combines to form spikes, and then there are two ways: whether these spikes are directly expressed on the surface of cells, like as you can see, they are directly cells expressed, or they are chopped down into small particles, small fragments, and these fragments are expressed in combination with the MHC molecules. MHC molecules, as you can see, they are expressed on the surface in combination with the MHC molecules. Now when this cell is infected by the vaccine particle, our immunity like innate immunity or adaptive immunity, sense the changes in the cell, that there is something wrong in the cell, and they cause the natural killer cells to activate, they cause other cells to secrete interferon, and they cause the immunity cause the phagocytes like macrophages to activate, and all these cells in combination with some other cells cause death of the virus infected cell, releasing the spike protein and its fragment into the surrounding. So how the spike protein and its fragment cause the activation of the T cell and B cell and formation of the memory T cell and B cell, we will discuss in the other slide. But before studying that, keep it in mind that the main protection of the vaccination is provided by the memory and the active T cells and by the antibodies, which are formed by the plasma cells, and the memory B cells, which are converted into the plasma cells. These memory are so important in the protection or immunity after vaccination because they are very specific B cells against a very specific antigen which we have been exposed previously, and they respond very vigorously and rapidly against this second exposure of antigen. And they are important because these memory B cells have a very low threshold of activation, i.e. they can be activated by very less antigen as compared to the first exposure. And the second one is that they produce much more antibodies than the new activated B cells. Now talking about the mechanism at how these memory and active T cells are formed. As we have already discussed that the infected cells are killed by the innate immunity like natural killer cells, etc. So they produce or release some kinds of protein like spike proteins and its fragments into the environment. These spike proteins and fragments are taken by the antigen presenting cells like macrophages and dendritic cells, and this antigen presenting cells has a unique type of MHC called MHC type 2. This antigen presenting cells present the antigen in combination with this MHC type 2 to the CD4 positive T cell, which is also called follicular helper T cell. This follicular helper T cells like this is activated. This is the antigen presenting cell, presenting the antigen in association with the MHC type 2 to the helper T cells. These activated T cells release different kind of cytokines, which results in the activation of macrophages, which kills the virus and virus infected cell. Virus infected cell results in the inflammation, which decrease the spreading of the virus, and activation and differentiation of T and B lymphocytes. In this T cells proliferation, some of T cells are converted into the active helper T cells, which in turn secrete the cytokines or activate the cytotoxic T lymphocytes, and other are converted into the memory T cells, which remain in the blood or lymphoid organs for so many long time. And this T cell provide the actual lifelong immunity. These T cells. We will study about the B cells later. Also keep in mind that every cell in the body except the non-nucleated cells has another type of protein, which is called MHC type 1 protein. And when any kind of cells are infected by the virus, they degrade this virus and present their antigen in combination with this MHC type 1 to the cytotoxic T cell, cytotoxic T lymphocyte or cytotoxic T cell. In this cytotoxic T cell degradation and killing of the infected cells. So now, as we have discussed the activation of T cells and the formation of memory T cells, now we are going to study about the activation and formation of B cell and memory B cells. So how to activate the B cells? We have two pathways. One is the T cell dependent pathway and the other is the T cell independent pathway. The T cell independent and this T cell dependent pathway. The T cell independent pathway, in the T cell independent pathway, the B cell are activated directly by the large macromolecules like capsular polysaccharides, lipids, DNA and RNA, etc. But the T cell independent pathway is short-lived. It is short-lived. Another pathway is the T cell dependent pathway, and in this the B cells are activated with the help of follicular helper T cells. In the T cell dependent pathway, the B cells are activated by the follicular helper T cells, and in this the immunity is long-lived, having long-lived plasma cells, and it is much more specific than the T cell independent pathway. Now, as we have already discussed that T cell independent pathway in which the B cells are activated indirectly, in which the B cells are activated directly by the microbe, capsular polysaccharide, DNA, RNA, lipids, etc. In this, when the B cells are activated, it results in the formation of antibody, which cause neutralization of the microbe, phagocytosis, i.e. opsonization, and the complement activation, which kill the microbes. But it is, as we have discussed, it is short-lived. Another pathway is through the helper T cells, in which the antigen presenting cell is usually the B cell, which present the antigen to the T helper cells and cause the T cell to activate, resulting in the formation of cytokines, which results in activation of macrophages and inflammation, and activation, i.e. proliferation and differentiation of T and B cells. Now we have discussed the T cells. Now we are going to discuss the B lymphocytes. The B lymphocyte differentiation and proliferation results in the plasma cells, which produce antibodies, and some of the B cells are converted into the memory B cells, which are much more specific than the plasma cells against the microbe or antigen. And this memory B cells react and respond to the second exposure very rapidly and vigorously, as we have discussed already. And these memory B cells are the main component of the immunity caused by vaccination.